Welcome everyone to this morning's MPG Primer. Today we are excited to have two speakers who will be giving back-to-back talks. And so I would love to introduce our two speakers today. First is Dr. Alicia Martin. She received her PhD at Stanford, working in the group of Carlos Bustamante. She then made the trip across to the other coast to do a postdoc here with Mark Daly's group at MGH and the Broad Institute. She is now an instructor in investigation at the Analytic and Translational Genetics Unit at MGH and the Stanley Center for Psychiatric Research at the Broad Institute. She will be giving the first half of the primer. The second half of the primer will be given by Dr. Omer Weissbrod. He completed his PhD at the Technion Institute in Israel and is currently a postdoc in Alka's Price's group at the Harvard School of Public Health. His work spans both statistical method development and applied research, and he is the first author of papers published in Nature and Nature Methods, co-authored papers in Nature, Science, Cell, and various other journals, and has attended many conferences in statistical genetics and machine learning. And so we are very excited to learn from both of them today, who excitingly, I think, are sharing a little bit of a peek into some coming science that they've been working on. So again, please post your questions as the sessions go on. And thanks again to our speakers.
Thanks, Sarah. Okay, so to get started, we're going to dive right into thinking about transethnic polygenic risk scores and what we can start to do to improve them and potential opportunities moving forward. So I'm a geneticist, a population geneticist, in fact. So as background, I like to start at the beginning. And for the beginning of polygenic scores, we really need to go back in human evolutionary history and think about how humans migrated, how they populated the globe, how they mixed along the way. So humans originated in Africa on the order of hundreds of thousands of years ago. And as they migrated into the Middle East, into Eurasia, and then into the Americas, each of these events resulted in some humans migrating forward, but some staying behind. So through each of these events, they took a subset of genetic variation with them, and these events are called serial founder effects. What that means is that the average out-of-Africa population has on average about 1 million fewer genetic variants than the average African population. So this human history over the past 100,000 years or so is really important to think about the landscape of genetic variation across the world, and is really important to think about when we're considering the design of human genetic studies. So if we've looked at the past decade of human genetics studies, it's been an incredibly exciting time to be doing human genetics. So we've learned a ton about human history, like I was just mentioning, through genetic studies, but also about the biomedical basis of disease from exponential growth over the course of the last decade or so. So this has been really exciting progress, and I can't imagine a more exciting time to be doing human genetics. This really serves our genetics mission of providing, biological hypotheses from unbiased genetic associations, which then feeds into providing the basis for downstream functional analysis and eventually, hopefully, therapeutic insights. But with that amazing progress, there's actually been a huge diversity problem. So now we're looking at the same trends over the course of the past decade, but colored by population or ancestry. And what you can see is that about 80% of these participants currently are of European descent, which is really far out of step with the global population, where European ancestry populations make up about 16% of the world's population. And perhaps even more troublingly, if you look at the bottom chart that shows this as a fraction instead of as absolute numbers, you can see that the representation of non-European ancestry participants peaked in around 2014, and since then has either stalled or slid in the other direction. So this is a big problem, because if we want to understand the genetics of disease, we need genetic variation. Like I mentioned just a couple of slides ago, we know that genetic variation varies massively across the world based on how your ancestors mixed, how they migrated, how they expanded and contracted over the course of human history. Additionally, if we have a narrower set of genetic variation that we're looking at, we have fewer shots on goal, so to speak. We have fewer attempts to actually find these genetic associations. So one of the really central questions that we've been asking is, how do ancestry study biases, these really big Eurocentric biases in genetics, actually impact the generalizability of our knowledge? And we're certainly not the only people thinking about this. This is a very hot topic right now. You've probably seen several of these headlines in very prominent media outlets, including things like the New York Times by one of yours truly, David Reich at Harvard. So there's clearly a lot of really strong interest in thinking about the generalizability. And there's quite a few different applications. So people are thinking about polygenic scores in the context of embryo selection, which is very sensitive and a little bit concerning and alarming to several of us right now. There are thoughts on how we can use polygenic scores in the context of educational attainment to consider social policy. So these are really, really touchy, particularly because the eugenics movement sort of spun out of the dawn of human genetics in general, and so white supremacists have kind of latched on to genetics in general. So we have to be really culturally sensitive about these topics. Okay, but more to the science at hand. When we're thinking about how ancestry study biases generalize, or ancestry study biases impact the generalizability of our knowledge, there are two really important considerations that we need to take into account. The first thing that we need to consider is that we think that causal variants are mostly shared across populations. This has been borne out in quite a bit of primary literature so far. So pretty much wherever we've looked, we've seen that causal variants look like they tend to be shared. There are a few exceptions to this, but they are very few as far as we know. The second, however, is that things like allele frequencies, linkage disequilibrium, environmental factors, and other more complex factors differ across populations as a function of one, population history, and two, current environments. Omer will get more into the genetics points later on. So when we're trying to think about this generalizability, one tool that we have in our arsenal is using a polygenic score. So I think all of you have probably heard of a polygenic score at this point because it's so highly considered in the media, but a polygenic score is really relatively simple. It's a genetic prediction of an individual's phenotype. So this requires that we have two study cohorts. We have a discovery cohort, a GWAS, a genome-wide association study that's been run previously, and we have a target cohort or a set of individuals where we're interested in actually predicting a trait. So when we have this discovery cohort, we take the effect sizes from that discovery cohort, we multiply them by the genotypes in our target cohort, we basically sum that up across the genome, and basically that's our score. There are a few more complex considerations to keep in mind. There are some fundamental choices, including what SNPs do I include? Do I just include all of the SNPs across the entire genome, or do I subset to some fraction of those? And then what weights do I apply? Do I need to make any adjustments to the effect sizes that were learned in that original GWAS? Other considerations are the fact that LD may or may not differ across the populations between our discovery set or our target set and across different regions of the genome. Additionally, there's variability in how reliable our signal is, which can be sort of tagged by the p-value in our GWAS. So this is something else that we can also sort of use as a tuning parameter sometimes for thinking about which SNPs to include. There are also several different methods for constructing a polygenic risk score. The simplest and probably most commonly used is pruning and thresholding, but better methods exist, including things like LD-PRED and SBASE-R. So these are varying degrees of complexity. So LD-PRED and SBASE-R are more model-based when thinking about things like LD. Pruning and thresholding is a bit more heuristic, but is relatively straightforward to apply. Okay, so there's been a lot of press about polygenic risk scores, and I've pulled a quote from a perspective that came out that we were really interested in reading that said, "We propose the time has come to incorporate genetic risk scores into clinical practice." And these authors are really well-regarded cardiologists at Stanford. They're really wonderful colleagues. And so I thought they made a lot of really good points, including the fact that previous criticisms of polygenic risk scores were mainly due to limited sample size that drove low statistical power and meant that polygenic scores just weren't that accurate. But we're overcoming a lot of these sample sizes now, as I mentioned at the beginning. And so we're actually getting to reasonably well-powered polygenic scores that in some cases are as useful or as informative as biomarkers that are currently being used in clinical risk models. Additionally, polygenic risk scores provide really cheap tests that provide insights into many diseases. So you can be genotyped, say by 23andMe or some other direct-to-consumer company. A genotyping array these days is less than $100, and you can learn about your genetic risk for many different diseases, for say type 2 diabetes or schizophrenia or obesity or cardiovascular disease or cancers. So there's tons of different things that you can learn about. Additionally, you would never consider a polygenic risk score on its own. You would integrate this with other biomarkers to inform clinical risk models and therapeutic decision making. So polygenic score on its own is never going to be the be-all end-all for determining whether or not you have a disease. There's always going to need to be some follow-up diagnostic and some integrative risk model is going to be necessary for public health purposes. So these are all really great points, but there's no discussion of ancestry in this perspective, and so we took sort of deeper dives into this. So I want to start at one of the points I mentioned originally, which is that causal effects are mostly shared across populations. This should be relatively intuitive if we think about the biomedical basis of disease, the fundamental cells and molecular pathways that go into these. They tend to be the same. Those pathways that break result in the same consequences no matter what your ancestral background. In GWAS, this has been borne out, for example, in schizophrenia. So when looking at risk of schizophrenia in European ancestry individuals and East Asian individuals, you can see this relatively complicated plot. Let's see, my mouse isn't working, sorry. But what you can see over here is that these lighter colors in this cream color and this lighter gray color are showing spots in the genome where the effect size is pretty much the same across the populations, but we just have differences in allele frequencies because of these differences in population history. In the red boxes that I'm pointing out, you can see that they're the two darker boxes, the brown ones and the dark gray, where we actually have heterogeneity in the effect sizes. So when you look at these plots, you can notice that there are actually very few of those brown and dark gray spots, indicating that causal effects are mostly shared across these populations. There are very few causal differences in schizophrenia. We've also seen very few when we've reviewed the literature for inflammatory bowel disease, BMI, lipids, type 2 diabetes, breast cancer, and so on. But how does this relate to polygenic risk scores? Well, when we're constructing polygenic risk scores, we're using correlated effects, not necessarily causal effects. So I like this little XKCD article highlighting the fact that statistics classes often teach you that correlation is not causation. So what does that actually mean for polygenic risk scores? Maybe this helps, well, maybe. So we wanted to learn more about this, and a few years ago, we took on this project to model human history and then attach it to complex trait disease models. And when we did that, we learned a few really key, I think, findings. One is that genetic prediction accuracy decays with increasing genetic distance between your discovery cohort and your target cohort. This is purely in simulation. We also had some empirical results based on sort of just observation and relatively small data. This was in 2017 when it was harder to get your hands on biobank scale data across multiple ancestries. So what this means is that if I have a huge pot of European ancestry data and very, very small pots of non-European ancestry data, the more genetically distant, thinking back to that map of human history, we are from European ancestry individuals, the worse we're gonna do at predicting any of these traits. The second point that we learned is that polygenic scores can differ across populations arbitrarily, but these biases are not necessarily meaningful. So normalizing across a global set of populations, on the right I'm showing you polygenic scores for height that we computed using the 1,000 genomes data. In this data set, we learned that European ancestry populations are predicted to be the tallest, followed by South Asian and admixed American, Hispanic, and Latino populations. And we predicted East Asian and African ancestry populations to be the shortest. So if we normalize across all of these groups, you can see that we're predicting that European populations are a lot taller than these other populations. We see a 5 standard deviation difference between the European and African distributions here. So thinking about that with some hard numbers, if the average European ancestry man is about 5 feet 9 inches tall with a standard deviation of about 2 to 3 inches, that would predict that the average African man would be shorter than me, shorter than 5 feet tall, which is clearly not the case, and defies all observation. The last thing that we learned is that neutral human evolution alone is sufficient to explain these differences. You don't need to invoke any arguments about natural selection to come up with these observations. So with those guiding principles in mind, a couple years later, we then looked in the UK Biobank to try to learn about how we're doing, to just benchmark this in real data. So we predicted several different traits, 17 different quantitative traits here, and looked at the prediction accuracy across these 5 different continental ancestry populations, and we normalized with respect to European prediction accuracy in an independent holdout set. So Europeans are standardized with prediction accuracy, relative prediction accuracy at 1, and across these different populations, you can see that we're doing much, much better in these European populations than elsewhere. So for example, we're doing about twice as well predicting European traits as East Asian traits, and about 4 to 5-fold as well in these Europeans than in African ancestry populations. We understand the genetic basis of this, but Omer is going to get into that a little bit more, so I will defer to him to explain some of that to you. Okay, we've also seen this sort of bearing out in different contexts across the human genetics literature. So for example, when researchers looked at schizophrenia and tried to use more diverse data to predict different traits, they've seen that rather than just using cross-ancestry results, if you have ancestry-matched GWAS, this is performing better. So for example, in the PGC schizophrenia data, they have some African ancestry GWAS in the left panel, some Latino ancestry GWAS, and then some meta-analysis in the last 2 panels. So when predicting in African ancestry individuals using the Discovery African Ancestry cohort, the target cohort on the left, this African ancestry cohort, has the highest prediction accuracy when you're using this ancestry-matched African ancestry GWAS. Consistently, in the second panel, you see that in Latinos, in the middle, we're doing a better job predicting in those with those ancestry-matched Latino data. So that's all fine and good. Ancestry-matched data gets you a little further than cross-ancestry data, typically, even when the numbers are considerably smaller. But on the right, you can see that the best results, the most accurate predictions are actually coming when we meta-analyze and combine these diverse cohorts. This is true for Africans, Latinos, and for European ancestry populations. So this is going to be the most meaningful way to predict all populations, not just those who are underrepresented, but it will disproportionately improve prediction for those who are currently underrepresented in human genetics studies. Okay, so moving towards like more recent research, I've been working with several African genetics researchers to try to understand how polygenic scores generalize on the highly diverse continent of Africa. These research fellows live in South Africa and Uganda and have taken on kind of this really big project to assess polygenic scores across different populations. We've revisited some of the work I did before, so this plot might look pretty familiar. This is a paired plot where we're looking at prediction accuracy relative to the European ancestry populations when we use only the UK Biobank European prediction data. So the UK Biobank is far and away the largest data set we're looking at here, where we have a discovery cohort for these 30 some odd different traits that we're looking at of about 350,000 individuals. And then we're also using Biobank Japan data, which has about 162,000 participants, and the Population Architecture for Genetic Epidemiology, or PAGE study, which has about 50,000 participants and includes African Americans, Hispanic and Latinos, East Asians, Native Hawaiians, and a relatively diverse set of individuals, all of non-European descent. So when we combine these data, the BBJ, PAGE, and UK Biobank data, we improve prediction considerably for some individuals. So you can see in the paired plots here, I'm showing you the different populations. On the left of each of them is when we just use the UK Biobank data to predict, and on the right is when we use the combination of all these data to predict. So we do pretty similarly in the Europeans, but we do a particularly better job in admixed American populations with Hispanic and Latino ancestry, in East Asian populations, and in African ancestry populations. And this makes sense and reflects the ancestral makeup and composition of those different cohorts that we're bringing in. So we're doing an especially better job with those populations that reflect that ancestry. Notably, there are actually some traits that we do a very, very good job in the African ancestry populations. So those outliers in terms of prediction that we're doing a much better job are white blood cell count and our mean corpuscular hemoglobin concentration. We just dove in a little bit more to try to understand what's going on here. And one thing that we found is that prediction accuracy outliers tend to highlight well-known, really well-characterized biology that's especially relevant to particular populations. So for mean corpuscular hemoglobin concentration, for example, we find outliers that correspond to genes known to confer hemochromatosis, sickle cell anemia and beta thalassemia, microcytic anemia, and hemoglobinopathies. So these are really clear biological signals that are especially relevant to African ancestry populations based on their environmental exposures throughout history where natural selection has had a chance to act. On the right, for white blood cell count, you can see some signals that are especially pronounced in these diverse ancestry populations that are not clearly present in the UK Biobank. So these include, for example, the Duffy blood group allele that's known to confer resistance to malaria, and this malaria resistance allele is not present outside of Africa. Just to end before I turn it over to Omer, another broad guiding principle that we've learned is that there are complexities of comparing across different populations and cohorts. We spent a lot of time comparing the Biobank Japan and the UK Biobank to try to understand how prediction works across different populations. And keeping the epidemiology in mind is really important for these prediction accuracy differences. So for example, the Biobank Japan had a hospital-based recruitment strategy and ascertained on 47 different diseases in the cohort. What that means is that people in the Biobank Japan are going to be sicker on average than the Japanese population at large. In contrast, the UK Biobank took a volunteer-based strategy from the general population. Due to this volunteer ascertainment, people that are participating in the UK Biobank have well-documented higher levels of health, wealth, and education compared to the UK population at large. So those are some considerations in mind that are challenging when we're thinking about multi-ethnic polygenic risk scores. With that, I'm going to stop sharing my slides now so that Omer can transition to his part.
Right. Let me share my screen. All right. Cool. Hi. So thanks everybody. So I'm Omer. I'm a postdoc with Alicia Price at the Harvard School of Public Health, and I will dive a bit more into the technical aspects of trans-ethnic polygenic risk scores. I mean, Alicia did a great job at introducing this, so thanks, Alicia. So hopefully by now you're all convinced that polygenic risk scores show a lot of potential at identifying individuals at elevated risk of disease. And there's been a few high impact papers lately and mainstream media coverage. So this sounds promising, but as Alicia said, right now we have these potential health disparities because on the one hand, most of our genetic data consists of Europeans, and on the other hand, when we try to train prediction models using Europeans and then to apply the prediction towards non-Europeans, then we get this huge drop in accuracy. And this is a problem. This is, of course, a very serious clinical problem, but it's also, I think, a scientific problem in the sense that, you know, I think about these trans-ethnic polygenic risk scores as kind of stress tests of everything that we do in genetics. So this test our hypotheses and our assumptions, and what happens when our results don't generalize, because in science you should get results that can generalize across different settings and cohorts and so on. So I've really viewed this as a stress test. And so I think it's really important for us to try to figure out what's going on and what goes wrong when we apply these prediction models across other cohorts or other ancestries than the ones they were trained on. All right. And so what I'm going to do in this talk is dive deep into what's going wrong. Why do polygenic scores or PGS lose accuracy across populations? And to make it simpler, so let's think about this cartoon figure where we have a discovery population and a target population on which we want to apply predictions. And let's say that we have these two SNPs, SNP1 and SNP2, and this trait, and SNP2 is causal for that trait. All right, cool. So I'm going to speak about 4 different reasons for what's going wrong. And the first is LD differences, which I think is what most people think about. And so let's assume, for example, that SNP1 is in LD with SNP2 in the discovery population, but not in the target population. So that means that we can use SNP1 to predict the trait in the discovery population, but not in the target population. And that's, of course, a problem. But you should notice that this is only a problem because SNP1 is not really causal. So we're not using the causal SNP to predict. And that's obviously suboptimal. So keep that in mind. The second reason for this loss of accuracy is MAF differences, minor allele frequency differences. And this is a problem even if we use causal SNPs. So for example, if SNP2 is rare or maybe even absent in the discovery population, but it's common in the target population. So even if we knew that this SNP was causal, that still wouldn't help us because we can't learn anything about this SNP if it's absent in the discovery cohort, right? So this is a problem even if we know the identities of the causal SNPs. And this is actually a pretty common scenario because we know that many causal SNPs are specific to only one population, and the gap is greatest between probably Europeans and Africans because of the out of Africa bottleneck and so on. All right. The third reason is more complicated, and that's different genetic architectures. So, you know, we always use these linear models to predict phenotypes. So maybe under the best, you know, linear model that we have, SNP2 has an effect of plus 0.2 on the trait for people carrying the minor allele in this population, but it has minus 0.1 in the target population. And why does this happen? Probably because our models are wrong. So we always assume these very simple linear models, but in truth, we probably have nonlinear effects coming from G by E, genotype by environment interactions, or G by G from, you know, genetic interaction between variants. And so that means that basically our linear models are approximations that are population specific, that don't generalize well across different populations. And that's, of course, a problem. And the technical name is trans-ethnic genetic correlation. So if the trans-ethnic genetic correlation is smaller than 1, that means that we see different effects in different populations. And the fourth thing that would go wrong is different heritabilities. And this could happen, for example, if we have some population-specific environmental factor. So maybe there is some environmental factor, maybe some diet in the target population that affects the trait. So maybe the trait is, I don't know, blood pressure, and there is some diet that affects blood pressure, and suddenly this becomes the main determinant of blood pressure. So suddenly genetics becomes less important, right? And if genetics becomes less important, then of course that means that we can predict as well as we did in the discovery population. All right, so we have these 4 different reasons, and I think these, the union of these covers most of what could go wrong. But I want to emphasize that these are not mutually exclusive. So that means that, oops, sorry, these are not mutually exclusive. So that means that math differences, for example, lead to different heritabilities and so on. So they're kind of confounded with each other, but the union of these things, I think, covers most of what could go wrong. All right, so I'm going to briefly mention I think the 2 strategies to mitigate this loss of accuracy. So let's go over these reasons one by one. So first, I spoke about edit differences. And so conceptually, the solution is simple. We should just try to predict using causal SNPs instead of non-causal SNPs, because we wouldn't care about these edit differences if we knew the identities of the causal SNPs, right? And of course, it's easier said than done. This is a very hard problem, and it's related to fine mapping, and I'll expand on that in a bit. But at least conceptually, if we knew the causal SNPs, we wouldn't care about these LD differences, which is important to understand. So that's not going to help us with the other reasons, though. So if we have these math differences, where we have a SNP that is rare or absent in the discovered population, or if we have these different additive genetic architectures, then we simply can't learn what we need to learn from the discovered population. We have to use data from the target population. And then the question becomes, how do we optimally combine data from both populations? Because at least nowadays, we have so much more European data than non-European data that it would be really wasteful to not put this data to some use to improve the predictions for non-European individuals, right? And so this is just, the broad strategy, and these are two open research questions that we as the genetics community should tackle. And as a shameless plug, this is stuff that I'm working on, but I think this is a general strategy in any case. And I didn't speak about the fourth reason. What can we do about it? Nothing. And that's because it's not really a problem. If the heritability is different, then it's different, and there's nothing we can do about it. So no strategy here. All right. So before speaking about these in more detail, I just want to point out that there is a very nice preprint out there that gives us a very nice principled quantitative framework to figure out how these factors contribute to the loss of accuracy. And so I'm going to go over this very briefly just to give you a taste. So this preprint shows that the relative accuracy, meaning the ratio of the R square between the targeted discovery cohort, can be given by a product of several terms. So one is the squared trans-ethnic genetic correlation, which I spoke about. The second is the heritability ratio. The third is the ratio of the variance of the polygenic scores. And the fourth is kind of complicated, but I just mentioned that there is a component of the similarity of the LD between the two cohorts, and another component of the math similarity, and another component of the baseline PGS accuracy, and they all mix together pretty nicely. So it's a very nice principled framework to figure out different factors contributes to this loss of accuracy. So this is very nice. I'm not going to go into the details. I just want to mention that the authors of this preprint use this framework to try to figure out why we lose accuracy when we apply polygenic scores from Europeans on non-European populations in the UK Biobank. And they reached the surprising conclusion that LD and math differences explain most of the loss of accuracy in African populations, but not in South or East Asian populations. And that's what you can see in this figure. I don't think I have the time to go into the details of this figure, but just in a very broad nutshell. So they constructed two prediction models. One considers only LD and math differences, and the other considers the other factors as well. And the simple model did a pretty good job at predicting the African loss of accuracy, but not at predicting the South and East Asian loss of accuracy. All right. So what I'm going to do in the remaining time is to speak a bit about each of these factors in more detail. And I see there's a question. Yeah,
Do you want to interrupt you with it? This is from Anna Lewis. She was wondering if the lack of solution to environmental differences, if you think that those people who are working on clinical implementation of a polygenic risk score should be sure to consider those environmental differences or not.
All right, that's
controlled for.
Yeah, that's a great question. The answer is a bit technical, so I don't think I have time to go into this in lots of detail. But when we think about accuracy, we think about accuracy compared to, the baseline levels of the trait. So if we have some environmental factor that is very dominant, then I didn't say whether genetics become less clinically important or just less statistically important. So they clearly become less statistically important. They might still be very clinically important because people, I don't know, who don't take this diet, some of them are still, suffer from some, health issues because of, their genetic dosage. So I think we should really tease these two things apart. It might be the case that even though our accuracy is lower, we could still identify individuals at risk and treat them. So yeah, these are two separate questions.
Thank you.
All right. So let me minimize this. All right. So I'm going to speak about each of these factors in just one or two slides. And let me start with LD differences. So LD differences are kind of easy to reason about because we can actually see them. We can look at genotypes of different people in different populations and look at the LD blocks. And we can see that there are substantial LD differences between different populations. So this is a figure from Alicia's 2019 paper. So at the bottom, you can see that when we compare the LD across different populations in the UK Biobank, we get substantial differences, for example, between East Asians and Africans here. And this corresponds to the population history model of the out of Africa bottleneck. So we know that Eurasians diverged from Africans at a very around, I don't know, 1,000 or more generations ago. And then at a later time point, Europeans diverged from East Asians. And so we can see that reflected in the LD of these populations. And we actually have, again, a nice principled quantitative framework to understand how these things work. So we know that LD similarity decays exponentially with the number of generations since two populations separated. And so here are just two examples of what our models show how the LD will be correlated across the populations with time. So you see that with every generation, the LD becomes less and less similar across two populations. This depends also on the recombination rate, but the bottom line is that we have a clear understanding that explains why we get very different LD blocks in some cases for African populations than for European populations. I mentioned before that we wouldn't care about this if we knew the identities of the true causal SNPs. And so this is related to fine mapping. So just a few words about fine mapping. So the idea of fine mapping is that it's a class of methods to identify causal SNPs. And the idea is that we take two things, we take GWAS results, and we take a matrix of LD, and we combine these two things together in a probabilistic model to estimate the causal probability for each and every SNP. How likely is this SNP to be causal? And this has very deep relations to polygenic risk score methods and models. So without going into the technical details, we can combine these two ideas so that we can try to predict using SNPs that are more likely to be causal. And I think this is the way forward to address these LD difference issues. Right now, most methods don't even try. So most methods just so they're very likely to miss the causal SNPs by definition. All right. So the second thing I want to speak about is math differences. And it says my internet connection is unstable, but I hope it's fine. So what do we know about math differences? So here are a few pieces of information from recent literature. So one thing that we know is that low frequency SNPs tend to be mostly population specific, and this makes sense because if a SNP is low frequency, it means that the variant happened only a small number of times, so it probably didn't happen across multiple populations. And so on the left, you can see a figure from Alicia's paper showing that we get lots of SNPs with low minor allele frequencies that are found only in one population. And on the right, we have a result from a recent preprint from Darsala et al., which uses population history models to argue that most causal variants for most traits are actually private, meaning that they're specific to only one population. And they argue that it's more common across Europeans. So we have more European-specific variants, and that's because of the recent population growth across Europeans. And this is a problem because if a SNP exists, if a causal SNP exists in only one population, then of course, then we can't use it to predict phenotypes in other populations. So we should just be aware of that and understand the dynamics of this pattern. In fact, this preprint makes the kind of bold statement that private variants explain up to 50% of the heritability of many complex traits. So this is part of a figure from that preprint. It's a preprint, so I'm not sure what's the latest word on that. It sounds a very high number, but I think that we can definitely say that population-specific variants explain a substantial proportion of the heritability of many traits. We also know that low-frequency SNPs have larger effect sizes than common SNPs because of negative selection. So on the left, we can see a nice figure from a preprint by Luke O'Connor, a paper by Luke O'Connor that shows that we think that we understand that low frequency SNPs can have large effects, but common SNPs can't have large effects, meaning that there is a lot of important variation that comes from these low frequency SNPs that might be specific to only one population. So this just explains partially why we get this loss of accuracy. And on the right, we can see a population history model showing that all of our models agree that SNP effects tend to be stronger for low frequency SNPs because of negative selection, because negative selection doesn't allow a SNP with a large effect to become common in the population. And how much of the heritability do these low frequency SNPs explain? So according to, I think, our most robust results, up to 20% of trait heritability. So here are two figures from two different papers that arrive at the same conclusion, one based on analysis of traits and one based on population history models. But this is a problem, right? Because this means that up to 20% of the heritability is explained by these SNPs that are more likely to be specific to only one population. And so this already places an upper bound on how well we can predict phenotypes in other populations. All right. The third thing I want to speak about is different genetic architectures. And this is a bit more complicated. So one thing that we think that we know at this point is that causal effect sizes are strongly but imperfectly correlated across populations. And a bunch of studies show that the typical correlation across the causal effect sizes of variants is around 80%, 0.8. So here is a figure from Alicia's paper that computes the trans-ethnic genetic correlation, which is a measure of the correlation between these effects across Europeans in UK Biobank and Japanese individuals in Biobank Japan. And you can see that there is a wide range of estimates with large standard errors, but I think that most of them are around 0.8, and 0.8 is less than 1, right? So this is a very robust result showing that our effect sizes are actually different across these different populations. And of course, this also places kind of an upper bound on how well we can predict using only one population. And here is the more recent result from a preprint from our group, which shows that causal effect sizes tend to be more population specific in functionally important regions, meaning that the trans-ethnic genetic correlation is lower in coding regions, for example. And again, this kind of places an upper bound on how well we can predict in different populations. All right. And the last thing I want to speak about is different heritabilities across different populations. This is the messiest out of these four things because estimating heritability is very tricky. It's kind of a can of worms, lots of subtleties that I don't have time to go into. But I'm going to show you a few results that give us a few hints about what we can expect. So on the one hand, we think that we know that there is a similar load of deleterious variants across populations. And this suggests similar heritabilities, at least for traits that are correlated with fitness. This is an encouraging result, but I'm going to show you some less encouraging results. So first, just a plain comparison from Alicia's paper again, which shows that UK Biobank shows consistently higher estimates of SNP heritability for many traits compared to Biobank Japan, possibly because of phenotypic heterogeneity, because as Alicia mentioned, the Biobank Japan was ascertained differently. But here are a few comparisons. You can see that there's a, I don't know, 30% difference in BMI in red here, and eosinophil count, there's a threefold difference between the heritability estimates. So this is just a result that we have. And in recent years, there is a very dominant trend of papers that show that probably many of our heritability estimates are confounded by environmental factors. So for example, here is a paper that shows that across different cohorts, there is a lot of heritability that we think is universal, but is actually specific to specific cohorts. Especially in this paper, they found that it is a problem for educational attainment and reproductive behavior traits, and less so for BMI, and also not at all for height. So this is one result that we know about. And going into more recent literature, just to show you many of the subtleties about heritability estimation, here is a nice result from a recent paper that shows that there's lots of evidence for heritability differences that is mediated by genotype by environment interactions. For example, we can see that we get very different heritability for diastolic blood pressure between men and women. And for BMI, we get very different heritabilities across different age strata. So heritability is larger for older people. And for years of school, we get very different heritabilities when we stratify by social economic status. So these are just a few examples for why when we estimate heritability, we may not be estimating what we think we're estimating. So we should just take that in mind. And of course, that could also be a problem for trans-ethnic predictions, because different people in different places probably have at least somewhat different environment. A very common trend is finding that BMI specifically, the heritability of BMI is strongly correlated with lots of different risk factors. There are many, many papers out there. Here are just two examples: weekly exercise frequency and weekly alcohol intake. So these are just a few warning signs, and let me sum up briefly. So I'm going back to this cartoon figure, and I spoke about the 4 different reasons for loss of accuracy. So LD differences, so we know for a fact that there are different LD patterns across divergent populations, especially between Africans and Europeans, for example, because of the ancient divergence time. We know that there are many math differences between populations, and that this is specifically a problem because many population-specific causal variants. Many causal variants are population-specific because of recent population growth. We know that there are different genetic architectures across different populations, so different SNPs have different effects. And we know that the genetic correlation is typically around 0.8. And in recent years, there is a lot of evidence for these G by E interactions. And different heritabilities, so this is more subtle. On the one hand, we know that there is a similar load of deleterious variants across different populations, but on the other hand, there is lots of evidence for environmentally mediated heritability, which could really complicate things. And this is really, an open research question that's being pursued. And I just briefly mention again the 2 paths forward, which I think is a call to arms to the genetics community. So if we want to, to do something about these LD differences, we should try to use causal SNPs and not tagging SNPs, because tagging SNPs by definition are, the tagging pattern is specific to one population. If we want to address the other 2 reasons, 2 and 3, we should really combine data from different populations. And Alicia mentioned some of the work that is already being done on that front. And that is it. Thank you. I'd be happy to take any questions.
Thank you so much, Omer, and also to Alicia. Those talks were really wonderful and had a really nice explanation of both the power and potential of polygenic risk scores. If anyone has any questions, please feel free to type them in the Q&A box or raise your hand, and we can always unmute you if you'd like to speak, to ask your question with your own voice instead of mine. I did have a question for both Omer and Alicia. As a clinician, I can think of how having, a risk-adjusted white blood cell count might be really helpful in clinical decision-making. Which polygenic risk scores do you think are closest to clinical use in terms of their accuracy and potential to improve care?
Yeah, that's a good question. Omer, do you want to take this or do you want me to?
So I think that the best evidence that we have right now is for cardiovascular disease. So there is a very nice paper by Amit Kara from Sekar Karitis's group that shows that we can identify individuals at elevated risk of heart disease. And the paper suggests that we can actually start giving these people statins and, maybe prevent this from happening in the first place years ahead, which is a very, promising result.
Yeah, I would add to that, the breast cancer and prostate cancer polygenic risk scores look really promising for a few reasons. One, in cardiovascular disease, fortunately, we already have several biomarkers that are already very informative. So measuring your LDL cholesterol, knowing whether or not you have hypertension, those are all really important things already. On the other hand, for a lot of the cancers like breast cancer, for example, we don't currently have clinical models that are very easily applicable to many people. So a lot of these are based on family history. The polygenic scores get a better tag on family history than, the documentation of a few of your close relatives. So those turn out to be more predictive. And so I'm holding out hope for those as well.
That's so exciting. We have another question that was asked, and it says that Alicia mentioned causal variants were usually the same across populations, whereas Omer stressed that many causal variants might be private to a population. There seems a bit of tension in those two statements, and we're just wondering for more of your thoughts on that.
So I think I can address that. I think that the solution is that the causal variants that we know about are common to both populations, but these are the ones that are easier to identify because they're common. And we're not speaking about the rare variants, which are less common, but they probably explain a large portion of the heritability. And these are probably much more likely to be population specific.
Yeah, I agree. I think the ones that are at the top of the genome-wide significant list where we can actually easily compare across populations, we have a pretty clear indicator that all of those are quite shared. On the other hand, we don't have a really clear idea of what's going on at the rare end of the spectrum, and most glances in so far show that maybe there's quite a bit more heterogeneity there than elsewhere.
And I'll ask one more question and see if any more come in. Is another pragmatic question. What screening would you want patients to have, and how often would you repeat it? Would it be a SNP array that's done early in life? Do you think that the technology is advancing often enough that the arrays would need to be updated frequently? How would you implement this in the population?
I think the implementation questions are hard to answer from a research perspective. So unfortunately, neither of us are clinicians that can directly address that. But I would say that since your DNA is stable, you do have some sort of lifelong information that comes out no matter when you're doing the genetic test, as long as you get a genome-wide survey. Although the discovery data that's going in is growing, day after day, and we're getting larger and larger data sets that are becoming more publicly available for many different diseases. So those will need to be continually updated for sure. But the genetic test that you take, hopefully you can take once or at least not many times as the technology evolves.
Yeah. I also addressed some of that. So we know that genotyping arrays are great for common variants, but we probably need to sequence whole genome sequencing to identify rare variants. And the question of how much rare variants contribute to complex trait heritability is under fierce debate right now. So we don't really know. Some people claim that they explain a lot of the heritability of common complex traits, and some people claim that they don't. So I stated the number of 20%. Some people claim that it's much larger than that, and we don't really know. So I think that the answer depends on the answer to that question. How much do they contribute to complex trait heritability? Of course, they're very important for Mendelian traits, but we don't know how much they contribute to complex traits.
Great. Well, thank you so much. Both of your talks were just wonderful, and I think set the stage for our MPG talk by Alka's Price's group, where we'll hear from 4 different projects. And so that will start at 9:40, so just enough time to make a coffee. And then just a reminder that this wonderful presentation will be available soon on the Broad YouTube channel as well for your review, should you have missed any of the slides. So thank you all, and we'll see you soon at 9:40.
Thank you.
Thanks, bye all.